solving today this exam paper from the latest session, March 24. We're going to also solve paper 6 exam after this. Let's start with the first question. They first asked you to define a catalyst. So that should be quite simple. You're only going to say it's substance, speed up reaction, right? So you're not going to talk about enzymes, you just talk about catalysts in general. And to be a catalyst, you should speed up reactions, but it would remain unchanged, right? Enzymes are like a special type of catalyst, but here they only wanted you to talk about catalysts. Now they're saying that enzymes are proteins. State and name the chemical elements that you find in proteins. Now, proteins are made of amino acids, but here they're asking you about proteins, which means what? Which means you need to talk about carbon, hydrogen, oxygen, the elements, and nitrogen. By the way, the only biological molecule other than DNA that contains nitrogen is proteins, right? Fats. Carbohydrates only have carbon, hydrogen, and oxygen. Okay, just gonna write all 4 elements. They gave you 1 mark for these and 1 mark for nitrogen. Okay. Next, they gave me that graph. It's a pH against enzyme activity. They said describe and explain. It's worth 6 marks. So what am I supposed to say first? First, it's the description. As we always say, you must take like an overview of the entire graph. So I'm going to say as simple as possible, say as activity increases and decreases. So that's make it simple, worth 1 mark if you just describe the overall trend. Next, we're going to say that there was no activity here and there was no activity here. So the activity was limited to the range between 4 and 10. You can say no activity at 0, at high or low pH. Okay. Next, I'm going to say here that the peak of activity was at neutral pH, at pH 7. Okay. So this was the description. As you can see, I've stated 2 general things. I specified also the highest peak here. Next, I'm going to talk about the explanation. You probably know that enzymes have substrates. The substrate would fit into the active site of the enzyme like this. Together, they'll make a complex. Now, if you exceed or go below that optimum pH, the enzyme will look something like this, right? The enzyme would be denatured. At that moment, the substrate won't be able to fit into the active site. So that's what I'm going to talk about here. So again, during the optimum pH, the enzyme would look something like this. Here or here, it would be completely denatured. So I say enzymes denature, the substrate won't fit into the active site. So you wouldn't be able to form that enzyme substrate complex. Okay. We're going to continue that question, or the first question here. They asked me complete the word equation for this reaction. It's lipase. As you can see, it's breaking down the substrate of lipase, which is fats. We're going to break it down into fatty acids and glycerol. You know that the activity of lipase requires another factor, which is bile. That's what the question is about. The next question is talking about bile. They said, while lipase alone won't be able to break down fats, so you would require bile. What's the role of bile? Bile's role is to emulsify fats. That is to break them down into small droplets. In addition, bile does such a very important thing, which is to neutralize the stomach acid, right? So without bile, the lipase would fail alone to work at its fast rate. You know, like bile comes from your liver. It's stored right here. It's like a pocket that the liver has. We call it the gallbladder, right? And then it's going to be poured into your duodenum, okay? So that's what I'm going to say here. I'm going to say bile is to neutralize stomach acid, right? It's alkaline. It's made in the liver, stored in the gallbladder, and it would be important to neutralize stomach acid because lipase would only work if the pH is nearly neutral, around neutral pH, okay? This means we're fully done now with the first question. I'm gonna now go to question 2, which is about the circulatory system. They said, state the name of the structures labeled A here and B. So they wanted you to label the valves that you have right here inside the heart. Now, valve A is, like the one on the left is the bicuspid. So always the left atrioventricular valve is the bicuspid, and the right atrioventricular valve is the tricuspid. The remaining valves here, valve B is the semilunar. Okay, so I'm gonna state A as either you can write bicuspid valve or you can say the left atrioventricular valve, and B is the semilunar. Okay, complete the sentence about blood vessel C. Do you see that blood vessel C? It's like here, right? That is C. C is the aorta, and the aorta is meant to take oxygenated blood all around your body, right? That's the aorta here shown. Let me show you what I have here. That's C. That's vena cava, right? Vena cava is the one that carries the oxygenated blood. And from here on top, I have pulmonary artery, and I have the pulmonary vein. Blue deoxygenated, red oxygenated. Anyways, here I'm just going to talk about the aorta. I'm going to say its job is to carry blood from the left ventricle to the entire body. Okay, and next they're showing me here a case of somebody who has a hole inside its septum. It's just like something like this. So that's the left atrium, that's the right atrium, and you have a hole between both sides. That hole could occur in the ventricles or between the ventricles or between the atria, but either way it has the same effect. What effect will this hole have? Usually you have here, as we stated earlier, here is oxygenated, right? And here on the right side is deoxygenated. But with this hole, the oxygenated and deoxygenated blood will mix up. And that's not a good thing at all. You know why? Because the blood now is going to be delivered to the body, and that blood doesn't have enough oxygen. So some of the blood going to your brain, to the body, will contain deoxygenated blood. So that's what they asked me about. They want me to talk about the effect of this hole on the transport of oxygen. I'm going to say the hole is in the septum, because that wasn't stated in the question, and it would cause the mixing of oxygenated and deoxygenated blood. So now the atria will pump less oxygen, and you will be delivering less oxygen to the entire body. So probably rate of diffusion will slow down. They didn't ask me about this, but maybe they ask you about it. So you tell them there'll be slower rate of diffusion. There'll be less aerobic respiration because less oxygen is delivered. Okay. The same question continues here. It's showing us somebody who's been exercising for 120 seconds, 2 minutes, and they show me how much is the percentage energy released by aerobic and by anaerobic. Okay, so you can, at any moment in this graph, if you add up the rate or the energy from aerobic to the energy of anaerobic, you realize it's 100, because that's the only way you can get energy in your body, right? So again, if you notice here, the energy from anaerobic, it's 80%, and aerobic's 20, and so is here. So always the amount of energy from either process, they will add up together to 100. Okay. Somebody is asking, what's atrium? That's an atrium, right? Like in the heart, the top two chambers, that's an atrium, that's an atrium. Plural is atria. Plural of atrium is atria. Anyways, yeah, going back to the graph, they wanted me just to describe it. So there's nothing really special here. I'm going to state like this. I'm going to say both aerobic and anaerobic, they increase at the beginning, right? So we have an increase for either, but which one of these is increasing faster? You can check that the anaerobic, the dotted line here, is speeding up faster, right? It's like showing faster increase. Now, anaerobic decreases afterwards. So like right after the peak here, it starts to decrease. And you can see at the end, they're equal right here, right? They intersect right here. So it's worth being mentioned. And lastly, you're going to say, well, they remain constant. So after the exercise or near the end of the exercise, they remain constant. I just want to explain to you how to describe. They didn't ask me to explain, but just like for our information, because it's quite important. Look, the reason why this athlete has increased his anaerobic, because at the beginning, the muscle would fail to get enough oxygen. Any muscle in the body, if you start exercise without warming up, you would find that the muscle can barely get enough oxygen. So the amount of oxygen delivered here is going to be little. The muscle will generate lactic acid. So you'd find that the lactic acid builds up inside the muscle. For that reason, the athlete would have somehow to take that lactic acid from his blood. It's going to be taken to the liver, and there it would be broken down. That lactic acid would be broken down aerobically. So the reason why the rate of anaerobic increase beginning because he did not have enough oxygen to his muscle. And the reason why aerobic increased here is because he is trying, after the exercise, he's trying to break down the lactic acid he built up in his muscle aerobically. That's what we call, you've heard of it, probably have heard of it, we call that oxygen debt, okay? So again, they said, write balance equation for aerobic respiration. So I'm going to say C6H12O6 plus oxygen. That's like the complete opposite equation to photosynthesis. So you could learn either equation, you reverse them, just make sure you write the correct equation for each one. So that's aerobic respiration. Flip it and becomes photosynthesis. State the name of the chemical that causes the oxygen debt. We said it's lactic acid. All right. This means we're done with question 1, question 2. We're now going to the third question. Third question talks about tropic responses in plants. So they took a plant like this and they put the plant sideways, just like the one I have here. You take it, you flip it sideways. As you do so, you know what's going to happen to the chemicals, the oxons? You'd find that these oxons will fall to the bottom side of the shoot, like this. Who's pulling them down? Gravity, right? Just like take any bottle, put it sideways. Gravity will pull these oxons downwards. Now you find that this side is going to grow faster than the top side. So the bottom side of the shoot will grow faster. And that causes, you know why I call this response? I call it negative gravitropism. Why negative? You call it negative because it's pointing the shoot against gravity. So for 1 mark, I'm just going to say gravitropism, but it's 2 marks, you would say negative gravitropism. I would always write negative gravitropism anyways. Explain how growth of the plant shoot is controlled. So I'm going to say that oxons, just like always, I say, where are the oxons made? I say, well, oxons are made at the tip of the shoot. They're going to be moving downwards to the bottom side. So you'd find them all here at the bottom. And oxons would cause those cells at the bottom, it would cause them to elongate, right? So that's why we have this side growing faster, this side growing slower, and the shoot is pointing upwards. You can't say anything about phototropism because this experiment is done in the dark, right? Usually, if you are to study gravitropism, you must turn off the lights. Otherwise, you wouldn't know what's the source of or what's the stimulus for the response. Okay. Same question. They're asking me direct question here. They said state 2 uses of water in plants. I'm going to show you all possible uses of water. You could use water in plants for cytoplasm, right? That's like where chemical reactions happen. You can state that water is how materials are transported in the plant, right? You may also say that water is needed for turgidity, because if there's not enough water, plant cells will become flaccid. You may also say water is to prevent wilting. More obvious to say it's for photosynthesis, right? So like many uses. I'm just going to state 2. In the exam, you're only going to state the 2 that you're most confident with. If you state any more than this, you're just going to ignore what's written underneath. So in my case here, if someone is marking my paper, he was just going to mark the first 2 correct, if they are correct. Okay. Next page, they said you got root length against time. So we're measuring the length of the root against time. And they said calculate percentage increase between 48 hours, right here, until 96 hours, until here. So let's read from the graph how much is the root length at 48 hours. It's right here, right? So it's like 9 units of length, millimeters. And here it's 25, I should say. So I'm going to find the percentage increase. How do you find percentage increase? You're going to state the difference, say 25 minus 9, and you divide by the initial lower number times 100. So here I'm going to say the difference divided by 9 times 100. They want it to 2 decimal points. So I'm going to say 177.78. Okay, good. Question 4. It's talking, just like what we've been talking about yesterday or the day before, we're talking about female hormones. So here they did not tell me what is hormone R, what is hormone S, but we have studied these hormones in order, the right order. So there's always FSH, estrogen, right? LH and progesterone, right? That's the correct order. So now I know that this must be estrogen and this must be LH because they peak in exactly that order I showed you here. So these are the names of the hormones right here. And then they said, state the day or days of the menstrual cycle. Where would you expect ovulation must occur? I don't really need to look at the graph because ovulation always occurs halfway during the menstrual cycle. So it's like here, right? If you start here, 28, 4 weeks, halfway, day 14. So we're going to say here is day 14. Shedding of the uterine lining, that's menstruation. So here they're telling me, where would you expect menstruation? Menstruation always occurs the beginning, right? So you can state any time here, right? Give a reason for how the concentration of progesterone would change during pregnancy. So again, we're now going beyond that cycle, and we're expecting that this lady from the graph here has got pregnant. How would you expect the concentration of progesterone to change? Now here, the graph here is for somebody who didn't get pregnant, but if she's getting pregnant, progesterone level will stay high for 9 months as long as she's pregnant. Why should progesterone stay high? Because this will ensure that the lining of the uterus stays thick. So take a look here. This is the lining of the uterus. As long as progesterone level is high, the lining of the uterus must stay thick, right? That's the job of progesterone. But who's going to make progesterone? Like usually, progesterone comes from two sources. It could come from the yellow body or corpus luteum in the ovary. That's during the menstrual cycle. Plus, during pregnancy, it's going to be the placenta who's going to make that progesterone. And so here, I'm going to say concentration should stay high during pregnancy to maintain the thickness of the uterus lining. And it's also important for what? You know, like progesterone stops or blocks or inhibits both hormones that come from the pituitary. Why should we block FSH during pregnancy? Because you don't want ovulation to happen. You don't want menstruation to happen during pregnancy. So that's one of the important roles of progesterone. So again, they asked me, where does it come from? We just stated it could come either from here, the yellow body, corpus luteum, or the placenta. So placenta. Good. Right, next page, they said, describe the roles of FSH in the menstrual cycle. Let me just quickly show you all hormones and their role. So again, where does FSH come from? FSH comes from the pituitary. So is the LH, right? FSH, LH, they target the exact same organ, the ovary. FSH's role is to stimulate estrogen, and its function is to mature as many egg cells as possible, usually one egg cell per month. On the other hand, estrogen and what do you call it, progesterone come from the ovary. Estrogen comes from the follicle cells, but progesterone comes from corpus luteum, yellow body. The targets here are the same: uterus lining, uterus lining. Always, every hormone stimulates the one that comes after, all right? So that's positive here. And every hormone blocks or inhibits the ones that come before, except progesterone blocks both hormones, FSH and LH, all right? So again, you would find that estrogen stimulates LH. LH stimulates progesterone. Progesterone is no hormone after progesterone. It's not going to stimulate anything else, all right? And what would inhibit is always the hormone before, right? So we're inhibiting whatever hormone comes before. Short for their function, progesterone's function is to build up, repair. Progesterone is to maintain. So there's a big difference. When I say estrogen, it's to repair. When I say progesterone, it's to maintain. There's a difference here between the two things, right? And in one word, LH's job is for what? LH's job is ovulation, okay? So I'm just going to state here, stimulates the maturing of egg cells for FSH, and it stimulates the hormone that comes after, okay? We're going now to question 5. Question 5 talks about plant reproduction. They showed you a flower. You can see that the petals are big, plus there's spots. Do you know what we call those spots? We call them the guidelines. It's like, this is like flower spotted. Those guidelines are quite helpful. Do you know why? Because they allow the insect to find its way inside the flower. Plus, usually large petals are quite helpful because they allow the pollinator, whatever insect is, to land on them like this, right? So going back to the question, I'm going to tell them it's an insect pollinated. First, it's having large petals like serves in two ways. It's visible, more visible to the pollinator, and it's like place for the insect to land. And these spots or the guidelines are to attract more insects, to keep the insect inside the flower as long as possible. Okay? They're asking me here, describe advantages, disadvantages of sexual reproduction in a plant. Let's state the advantages first. One of the main sources of advantage of sexual reproduction is meiosis. So the whole variation here comes from the fact that with sexual reproduction, you have gametes, and gametes come from meiosis. So that's why living things that do sexual reproduction, they show wide variety. They are showing variation. And with variation, you get adaptation. Living things that show variation are more likely to adapt to new environmental conditions. So like if the conditions change, it gets cold, gets hot, only the living things that show variation would survive. All right. Variation is also helpful to like getting protected from infections, disease. Usually, if some infection occurs, living things that show variation are less likely to become extinct. Some members would survive, right? And with survival, what would be the next step? It would be evolution, right? So living things like giraffes, because of their variation, they were able to survive and evolve as species. Finally, something more specific to plants is seeds. Plant seeds are very helpful because when the plant seed is dispersed, when it's been blown away from the parent plant, it would land somewhere away from the parent. And that's a good thing. You know why? Because it would prevent overcrowding. So not all the members or not all the offspring will grow in the same place. All right. So advantages, variation, adaptation, evolution, right? Seeds and their ability to grow in new environments. What about the disadvantages? The disadvantage is the fact that you got two parents. You would require two parents. So that sometimes is difficult, especially if there is one plant and this plant is isolated. Also, it takes a lot of energy. Time and energy are disadvantages. What do I mean by time? It takes long time, like for living things that reproduce sexually, to reach maturity and reproduce. And energy, because making of gametes takes energy. Think of pollen grains and how much energy it takes to make all these pollen grains. Okay? The same question continues, and they're showing me here plants grown in Europe versus plants grown in North America. They wanted me to find the average of these numbers, or the mean of these numbers. This is the what is it called? That's leaf size, how big the leaf is in either continent. So I'm going to add up these numbers and I'm going to divide by 5. Quite simple, right? So we add them up, divide by 5, you're going to get 301. And don't forget the unit again, cm squared, okay? It's an area, right? It's written here. What conclusion, what's the most obvious conclusion that you can come up with from this data? You would find that those growing in Europe have larger leaves, right? So that's what I'm going to say here. So say plants in Europe have a greater leaf area than those in North America. The same question continues, and this question is like one of the most important ones that come like almost in every exam paper. What is this about? They said those flowers have developed large leaf area. So how did these plants end up having larger leaves than usual? Let's think about it that way. Let's assume that somewhere in the past, we had these plants and the leaves were small, right? Something like this. One plant was born, you don't say born for plants, one plant was grown here and it had a mutation. That mutation made its leaves bigger than usual. Now, this plant here had a great advantage. You know why? Because it could absorb more sunlight, it could do more photosynthesis, right? And it's going to grow faster than all the other plants in its habitat. So what's going to happen? This plant will reproduce faster, and you would find many of it now growing. Do you know what I call what just happened to the plant population? This plant population has evolved. This plant population has become better adapted to its environment. So look at the order of events I'm going to write here about. I'm going to state mutation leading to variation, and that variation here would allow them to adapt, survive, and you survive long enough, then you would be able to reproduce, right? Reproduce, okay? I would always write that's like scenario or the order of events whenever there's a question about some new feature or like adaptation, evolution to environment. So I'm going to say first, there is variation among those plants due to mutations. Plants have larger leaves. have better chance to do photosynthesis because they absorb more light. So those plants would survive and reproduce. And this plant that has survived, when it's reproduced, is going to pass. Now, here's an important term. It's going to pass alleles. Why don't I say it passes genes, passes DNA? Because all members of the same species, they have the exact same genes. What makes a small leaf different from a large leaf? It's an allele. It's different form of the gene, right? So what would that lead to? That would lead to the population evolving. And what do I call this form of evolution? I call that natural selection, which means what? Which means that nature always picks the more adapted, the better adapted, the big leaves, because those would help the plant to survive. Okay, now it's question 6. It's the last question. What's this question here about? They said you got something called phytoplankton. They contain chlorophyll. Automatically, when you hear about chlorophyll, this means they're what? This means they're like small plants, right? Like the algae you study in eutrophication. So they said, how do these phytoplankton lower CO2 in the atmosphere? Now, usually anything that got chlorophyll is producer, right? And producers do photosynthesis. As they do photosynthesis, they'll absorb carbon dioxide. Suggest. When you hear someone says suggest here, this means what? This means it's about something you're not very familiar with, right? Something you've never heard of, probably. Something called zooplankton. But you can always figure out what is it about. Look, where's that zooplankton label? They said eating feces. Quite disgusting. But what does that mean? This means it is breaking down dead matter, right? Decomposing. So that's why I'm going to say here, I'm going to say they are decomposers, right? They are breaking down dead matter or waste, not waster, waste, okay? The question continues, same question. They're talking here about ecology now. They said to state names of 2 processes that add CO2 to the atmosphere. So what events or what processes increase concentration of carbon dioxide in the atmosphere? You got a biological process like respiration, and you have the one that has led to the rise in CO2, so much combustion, burning of fuels. Describe the effect of the environment of having too much CO2. This means they want you here to talk about what? They want you to talk about the greenhouse effect. They want you to talk about how levels of CO2 has risen recently. That CO2, originally, it used to be very helpful, right? It was, we call that the greenhouse effect, which is the fact that CO2 and methane both reflect carbon dioxide, reflect infrared. That's infrared here, IR for infrared. But too much CO2 has led to what? Too much of it is now re-reflecting a lot of infrared, leading to what we call the enhanced greenhouse effect. So first of all, I'm going to tell them that CO2 is one of those 2 greenhouse gases, in addition to methane, which absorb infrared, leading to what? Leading to what we call, either you can say global warming, enhanced greenhouse effect. And it's also helpful to mention the consequence, because that's what they said here. Effects on the environment. So I can say something like decrease in biodiversity, extinction of species. I can say droughts, floodings, melting of ice glaciers worldwide. Okay. All right. They also gave me a permit, and this is they said the label on the permit, label the trophic level with herbivores. Where would you expect herbivores to be found? Now, always the first block is for producers. So that's always the producers. What would you expect the herbivores to be? The herbivores will be here, right? Right after the, so I'm just going to put label. I'm just going to label it as the question number BI like this, okay? Or you can write herbivores, not a big deal, right? Okay. Someone's asking me like, how do you get floodings while it's global warming? Look, the weather cycles in the planet changes. So if you ask grandparents, we tell you like, we used to live somewhere where it's cooler than it is now. They may tell you it wasn't raining that much. So all the weather cycles worldwide are changing. It's not like about just one pattern. No, it's like completely getting messed up. Even that next question is super important. It said, explain why pyramids of energy usually have fewer than 5 trophic levels. First of all, like we have 3 types of pyramids. We have the ones that we call pyramids of number. Those count the number of individuals at each trophic levels and is likely to get inverted like this, okay? So that's a pyramid of number, which is the worst one among them, like the very misleading, because as you can see, the tree shows here as smaller block than the rest. You also have what we call pyramids of biomass. Here, we're actually measuring the mass of each trophic level, and it would show something like this, right? Usually they're always upright. There's no way a tree will weigh less than caterpillars. What could be even better than the pyramids of biomass is if you could measure the energy. If you measure the energy stored at each trophic level, that would be like the best case. You know why? Because no way that the energy is going to be less in any trophic level than the plants themselves, because plants have the source. Plants get all the energy and they pass it on to other trophic levels. Why is this better than the pyramid of biomass? Because biomass might be misleading. Sometimes you may measure parts of the animal that are not edible, all right? Or maybe that wooden part of the tree that nobody eats. So all of this is biomass, but they're not contributing to the energy. So that's why if you could measure the energy stored at the trophic level, that would be the best way, the best representation of the flow of energy. So anyways, let's first answer the question. Let's explain why pyramids of energy usually have fewer than 5 trophic levels. Why is it that you cannot exceed like number 5 when it comes to any food chain? Usually, as you can see here, every time I go from one level to another, I waste 90% of the energy. Why do we keep wasting energy that much every time we go from one level to another? That's because of respiration. That's because of excretion. Living things respire and waste heat energy, excretion, and in addition to movement. That applies more to animals. So those 3 processes, they're life processes, but they waste a lot of energy. So that's what I'm going to say here. Energy is wasted by either one of these processes. 90% is wasted, 10% is passed on, right? So that's why very little energy is left. Like if I have any other trophic level here, like a hawk or owl or whatever is eating that cat, the population will be too small. And that small population wouldn't be able to support any extra trophic level. Here they say like 2 advantages of measuring energy rather than measuring biomass. So I can say with pyramids of energy, you can measure exactly how much energy is transferred rather than just measuring biomass. With biomass, as I told you, so much of living matter is not edible. So it's not going to be wasteless, it's not going to be realistic. I measure the bones while nobody eats those bones, okay? And that's what I'm going to say in my next point here. I'm going to say not all biomass has the same amount of energy, right? So for instance, carbohydrates will have way more energy than, for instance, calcium or other minerals you find in bones, for instance. Next part, it's a definition. What's meant by sustainable resource? Now, the word sustainable, like fish stocks, stands for what? Stands for something that you could make and it would never get used up. Usually plant products, like if I make a biofuel, it's 100% renewable, right? Because there's no way that we'll run out of plants. As long as you have air, as long as you have the sunlight, you'd always have plants. Even applies to bioplastic, right? You make plastics from plants, that's also sustainable. So generally speaking, any sustainable resource is the one that is produced as fast as it's been used, right? Here I should say as rapidly as it's used, meaning if I'm getting my products from a plant, the plant will grow more of that product and will not run out of it. Okay, getting to the last page, this paper, they said fishing can manage to promote conservation. Explain one way that fishing can be managed to conserve fish stocks. How many ways you can mention here? You can mention the net size, making the nets as small as possible so you don't kill the small fish. Also to build fish farms because you can conserve them, you can allow them to breed. You can also have fishing seasons, meaning you would only fish when you allow fish to breed, like maybe in the spring, and then you fish in the summer. So you ban fishing while the fish are still breeding. Also, you can restock fish, right? So you can breed them somewhere safe and put them back in their environment. So one more time, I'm going to say something like control the net size. You can say education. We didn't mention education, but it's like the easiest and the most obvious. If you educate people about the importance of fish, then they will avoid overfishing. And you can also mention closed season. What's a closed season? Somewhere where you ban fishing. Tell people you can't fish between Jan and March, for instance. And you build fish farms. So you allow fish to breed right there. Okay? And this means we're done with paper 4 for today. I'm going to solve one short paper 6 exam here. It's taken from 2019, this variant. It starts with the usual thing. They're telling you here about food tests. They said, describe how ethanol could be used to test a sample of fats. So as if someone is asking you here to do the emulsion test. So how do you do it anyways? You take the fats, right? You dissolve them in ethanol. You shake them well, right? And then you add water. If there are any fats, what would you expect to see? You would expect to see a white emulsion. So first I dissolve, I mix well, and then I add water. And if fats are present, you get an emulsion. The size of the emulsion tells you how much fat is present. Thicker, then there's more fat, okay? So mix with ethanol, add water, and white emulsion appears. Next, they're asking you, they did that Benedict test. They add Benedict, and then they mixed. They ask you, like, what are the next steps you're meant to do? So again, right after you mix and add Benedict, you're meant to do what? You're meant to heat in a water bath. Usually heat at 80 degrees Celsius. And then you will be able to see the results, right? So here I'm going to say heat in a water bath. Describe positive for reducing sugar. So you can mention any of the colors. You can say green, yellow, orange, red, blue. No, not anything but blue, okay? And then they're talking about proteins here. They did an experiment. Now, the most important line of any experiment is either the first or the second. Here's like the second line. They said all where it matters. They said investigate the effect of acid on the breaking down of proteins. So what's your independent variable here? We're studying the acid. So your acid is your independent. That's the one you're changing. And the breaking down of proteins by protease is what you're measuring. So we got 3 test tubes, P, Q, and R. P got water and substrate. Okay, so it's nothing. That's a control. That's the one we're going to use for comparison. Q has what? Has enzyme, water, and substrate. And the last one has enzyme, acid, and the substrate. Okay, so basically, they want to see how does the acid here in R affect the rate of digestion. Okay, so here's my observations first. Here, they said P stayed purple, Q is purple, and R is blue. What does that mean? With the Biuret test, if the color stays purple, this means what? This means there was no digestion. The protein is still present, okay? Same here. I did not get any results, again, with the enzyme alone. So you can see the protein is still present. But with the last one, I did get a result because the protein got digested. The purple color of Biuret test has disappeared. So what does that tell you about this enzyme? This tells you that this enzyme works best at acidic conditions. Does that remind you of any enzyme you've seen? There is like pepsin from your stomach works like at acidic conditions. All right. So what am I meant to do here? I am meant to make a table to show these results. Okay. So in any table, I need to put first the independent variable, which is the acid. We're going to put it right here on the left. And I have 3 test tubes, P, Q, and R. And there on the right, I'm just going to stick the colors. So I'm going to say purple, purple, and blue. I'm only meant to take these results here, right? If they said both tables, I would have included the, like, for instance, the type of solution I have, okay? So again, what's the most obvious conclusion here? Don't go very fancy with the conclusions here because this paper 6 is for core and extended, right? So make it as simple as possible. What could be like the best conclusion I could write here? I can say, well, when there's an acid and enzyme present, you get digestion. Or you may say, like, proteins only get digested if there's enzyme and acid. Or you may say, enzymes alone don't digest the protein. You must add an acid. But what's the point of that test tube here? Like, why did we have a test tube? We know in advance it's not going to work. I know in advance if there's no enzyme, there's no digestion. So why did we include that test tube here on the right? This test tube is quite important. This is what we call a control, right? So that test tube is helpful for comparison. Anyways, they said identify the variable that was changed or the independent variable in this investigation. Usually, look, in any investigation, the independent is the one I change, all right? The dependent is the one I measure, okay? So once again, what did I change in this investigation? I changed the acid or the enzyme, but it's more likely to be the acid, right? And what's the purpose of P? We said that P is to, like, comparison, right? To show me that the substrate, the protein, doesn't get digested by itself, right? Because what if this gave me like a blue color? Then it would have shown that there's something wrong with my setup, right? So just why 1 cm cubed of water was added to test tube Q. Why did we add water here? Like, why did we include water in this experiment? If I didn't add water, I would have had less volume here, right? And then it wouldn't be a fair experiment, right? It wouldn't be fair because one test tube would have less volume than the other two, right? So just meant to make like all the sets the same, fair comparison. And then they said, explain why there's a potential source of error in this. This student used only one syringe. Why is this an error? Why would this mess up like the whole experiment? Usually that's what we call cross-contamination, meaning that you're mixing solutions with one another because you're using the same dropper or the same syringe, right? Again, if I'm using the same syringe, then the error is going to be cross-contamination. And the effect is that you would put some acid in the first test tube or the second test tube, or you may put an enzyme in the first test tube, right? So for all of these reasons, that would completely make or mess up the experiment. Then they said, find one potential safety issue in this experiment. Now, you can see that we've used an acid, and acids are corrosive, right? It could cause skin damage. So that's what I'm going to say here, okay? Moving to the second question, they gave you an artery, and they wanted you to do what? They wanted you to draw that artery. And as always, when you are to draw whatever, you need to mind those 4 marks, which are, first, detail. What could be the detail point I could score here? I count the number of layers. So I count to see, well, there's one layer here, there's another layer here, right? There's another one on the outside, okay? Then I make sure that it's an outline, meaning I'm not going to shade that. It doesn't like make it look artistic and shade those areas here. And proportions, meaning you can see here that this side is wider or longer than the other side, right? I keep making it oval. I'm not going to make it a circle. And size-wise, you have to make it as big as the space that you have been provided, okay? So going to occupy as much of that area I was given. Again, I'm going to grab my pencil here, and I'm going to draw an outline, same orientation, okay? And I'm going to take that inner layer and make it like curved, right? And I'm going to include that inner layer. Do you see that part here? I'm going to also include it. That will give me the mark for the detail. It looks like this, all right? Something like this. And I can include something like this area here and this one here, right? Again, I did this before because it takes more time, and you would include that part here, okay? Okay. They did not ask me to label and not to draw any cell. Next, they said, find the length from A to B. So take your ruler and find that length from A to B here, whatever the length is on your paper, okay? And then record that length right here. So in my case, it's like 8. I'm going to write like 8 centimeters or 80 millimeters. I always prefer millimeters because it's easier to calculate rather than dealing with decimal points. And then they said, find the actual. So my image was 80, right? The magnification, which is given right here, is 13. So how much is the actual? I'm going to say it's 80 divided by 13. Let's just work it out here with my calculator. 80 divided by 13. It's 6.15. So I'm just going to round it up to 6.2. Okay? Keep the same unit as the one you use here, okay? Here they said, describe one similarity, one difference between the artery and the vein. So again, here you're only meant to see like visible similarities. You're not going to talk about valves. You're not going to talk about pressure. You're only meant to talk about what you see here. You don't mention oxygenated, deoxygenated blood because none of these things is visible. So the most obvious and the easiest thing you can say is size, right? You can tell that one of them is bigger than the other. You may describe the shape. You may tell them that one of them has more layers than the other. Okay. So here I stated similarities. Both have several layers. Both got lumen. Differences: smaller, thicker, narrow lumen. Okay. And next, they gave you a table, and it's about some athletes before exercise, during exercise, and after exercise. They're measuring their pulse rate, all right, neck or wrist. And they said calculate percentage increase, just like last time, percentage increase for pulse rate between 6 and 12. So again, I'm going to apply that same formula here, the difference over the initial times 100. So the difference is going to be 160 minus 78. And I'm going to divide by the initial, which is the 78, times 100 gives you the percentage increase. Okay. Next, they said plot a line graph on that grid to show time against the pulse rate. So I'm going to plot this here versus this one here. Okay. So let's put the time on the x-axis. Always the left column is your x. Okay. And the right column is your y. So the left column starts from 2 all the way to 18. So I'm going to put 2, 4, 6, 8, 10, 12, 14, 16, 18, time / minutes. Okay. And the y-axis ranges from 78, I'm going to like nearly 80, all the way to 160. Okay. If I start from 0, I'm going to end up in trouble here because I'm going to like leave all that section empty, and I will end up just with using that area on top. So I'd rather like put a line like this and start, for instance, from maybe 60 here, 80, 100, 120, 140, 160. Okay. This means I'll occupy as much of the grid as possible. And before I forget, I'm just going to copy the y-axis here. Okay. Next, I'll plot. I have 2 with 78. I'm going to do it quickly, roughly here. 4 with 78 as well. And 6, 78. And 8 with 125. I think I shifted that thing here. 8 with 125, which is here. And 10 with 148, which is here. And 12, 160. 14, 154. That's here. And 122. And last one is 94, which is right here. From here, what am I meant to do? I'm going to grab my ruler again, and I'm going to draw straight lines from here, right? There's no, if we start from the origin here, it will be completely wrong. So don't start from the origin because you don't have a result there, right? So that's my second one, right? Like this. All done with the rule. I did not do any smooth curve because that's how it's meant to do, okay? Use your graph to estimate the pulse rate of the person at 15 minutes. So let's take 15 minutes here, draw a line at 15, this, and draw another line like that. So it's going to be 140. Okay, so 140 units is beats per minute. Beats per minute. They wrote it for me anyways. And describe the results of the person in this investigation. So how am I going to describe this? I'm going to state like first, the pulse rate stays constant. Then we had like rapid increase. We reached the maximum at 12 minutes. It was 160. And then it decreased. So before exercise, we had constant pulse rate at 78. Pulse rate increases rapidly during exercise. It decreases after exercise, but it did not reach back to the resting, right? If I wait, I would have probably returned back to the 78, but based on my results, we did not return back to the resting. Okay, next and last is this one. They said plan investigation to show how running at different speeds affects pulse rate. Okay, always with these questions, I ask myself 4 questions. I like plan before I write. I don't write unless I know exactly what am I going to write and in what order. So again, they said here, different speeds and pulse rate. So what's your independent? What are you changing? I am changing the speed. What am I going to measure? I'm going to measure the pulse rate. What am I going to control? I can control things related to the athlete, right? Maybe the age, their gender, their lifestyle, right? Smokers, non-smokers. And I have to repeat. I repeat like usually 5 times. And any safety. Here it could be wise to make sure that whoever is participating is not like having heart problems, right? He has heart problem and I make him run, he might be in trouble, right? So any safety related to heart disease or something, okay? So let me show you my plan. I'm gonna take same person and I'm gonna make him run at different speeds. So one time I make him like jog or walk, and here I make him run. And before I repeat, like I'm gonna repeat this as I told you 5 times. It's very important to measure his resting pulse rate first, because I want to know exactly how much is his original pulse rate, and then I make him run, rest every time, and measure the pulse rate after exercise. So you really need to measure the pulse rate immediately after exercise. All right. So I'm gonna say first, measure resting heart rate, allow him to run like at high speed. That's my first one. Okay. And then measure the heart rate immediately. How do you measure pulse rate? You would measure pulse rate either the neck or the wrist. Now I need to let him rest. So give him 5 minutes to rest or more, and then ask him to run the same distance, but less or low speed. And again, measure the heart rate and find the difference. Okay. This means we're fully done with what we're meant to do today. I'm gonna let you go now if you don't have questions.